Hello friends, welcome back to my YouTube channel Astin Padasale. So this is a part 2 video. Currently we are discussing top 100 expected questions for your GATE Biotech. First question is pro-apoptotic BH3 only protein includes BAD, BIN, BIT, PUMA and NOXA. In the comment section you need to tell me what about anti-apoptotic protein and what about pro-apoptotic BCL2 only protein. Next thing, which type of signaling depends on local mediator that are released into extracellular space and act on neighboring cell. So the signaling, this type of signaling is called as paracrine signaling. Endocrine means you need to remember the keyword bloodstream, and synaptic means you need to remember the word neurons. Next question is, cancer cells often produce extracellular signal that stimulate their own survival and proliferation. This type of signaling is called as autocrine signaling. So your cancer cell and immune cells will be coming in a category of autocrine signaling. Next question is, digitalis, which is also called as digitoxin, is isolated from foxglove plant, and you need to remember the scientific name also. It is an example of which type of secondary metabolite. So it comes under the category of cardiac glycosides. The next question is, antibiotic that block the transcription in bacteria includes your actinomycin D and rifampicin. So they generally inhibit the activity of RNA polymerase. Next question is, which antibiotic block the translation in bacteria by binding to its 30S subunit include aminoglycoside, nitrofurans, pectinomycin and tetracycline. So you need to know about which type of antibiotic that block the translation in bacteria by binding to 50S subunit. So you need to remember whether they are asking about bacteria, whether they are asking about eukaryote, and next thing you need to remember whether they are asking about 30S subunit ribosome or 50S subunit, because that differs. Next, which protease enzyme that cleaves the C-terminal side of your aromatic amino acid? So it is your chymotrypsin, C for chymotrypsin, and it cuts at C-terminal side. Next question is, name the drug that inhibit the microtubule polymerization. So both your colchicine and colchimine, they inhibit microtubule polymerization. In other words, colchicine is a microtubule destabilizing agent. So they are involved in destabilizing your microtubule. So just read about which drug inhibit microtubule depolymerization. So don't confuse with that word. If you are asking the drug that inhibit microtubule polymerization, means the correct answer is colchicine. So it is acting as a microtubule destabilizing agent. Next, an antibiotic that is isolated from streptomyces that inhibit the flow of electrons from cytochrome B to cytochrome C1, which means we are asking which antibiotic inhibit complex number 3 in electron transport chain. The correct answer is antimycin A. So just remember, or just try to know what are the other antibiotic inhibiting your electron transport chain, because each complex is inhibited by different type of antibiotic. Just try to know, and also just try to read about uncouplers. Okay. Next, which organism is employed for phenol disinfection coefficient test? There are two organisms are employed. First one is Staphylococcus aureus, which is a gram-positive bacterium, and next one is Salmonella typhi, which is a gram-negative bacterium. Next, the two genes, sorry, it is GEMES. Name the two genes that are inserted into GM mustard. So GM mustard is a very hot topic currently going on in biotechnology. So try to know more about this GM, genetically modified mustard. Okay. So the two genes include your Barnase and bar star. These two genes are isolated from soil bacterium called Bacillus amyloliquefaciens. Okay, this Barnase gene will be conferring male sterility, and bar star will be restoring the fertility. Okay. Next thing is called as hybridoma technology. So in hybridoma technology, myeloma cells, they don't have HEPRT. So they are HEPRT negative. So in hybridoma technology, you need to know about HAT medium. Specifically in the HAT medium, you need to know about aminopterin. So aminopterin, whether they are blocking salvage pathway or de novo pathway. So you need to answer me in the comment section. Next, which RNA serves an adapter between your mRNA and amino acid during protein synthesis? I think you all know the answer. It is your transfer RNA, tRNA. Next, name the bioactive gibberellin. Okay, so bioactive gibberellins include GA3, GA4 and 7. 347. Okay, just remember the word 347. Next question. An organism that live under elevated hydrostatic pressure is called as piezophile. Okay, so you need to tell me what is mean by xerophile, what is mean by halophile, and what are the example of halophilic organism. All those things you need to tell me in the comment section. Next question, a strain of mouse that is genetically identical in all locus of chromosome except for a difference at a single genetic locus. It is called as congenic mice. So what is mean by syngenic mice? You need to tell me. I had asked many questions, both in my part 1 video and part 2 video. I had given you a lot of questions I asked and you need to reply me if you want to score somewhat good mark. Next question is, lipid soluble secondary messenger located on inner side of plasma membrane is called as your DAG, diacylglycerol. So your CAMP and Ca2 plus are water soluble and they are present on your cytosol. Whereas this diacylglycerol, they are located on the inner side of your plasma membrane and it is a lipid soluble. Next question is, which Vir gene function as a single stranded binding protein and protects your tDNA from the attack of nuclease during strand transfer? It is your Vir E gene. So it will be function as a single stranded binding protein. Okay. Next question is, allergic contact dermatitis is an example of. So it is an example of your type 4 hypersensitivity reaction. Other name for type 4 is your delayed type hypersensitivity reaction. So just know the example of type 1, type 2, type 3 and type 4. Okay. Next, genes that arose from a speciation event are called as orthologous gene. In other words, this question will be asked from genes that are arose from the common ancestor or from a vertical thing is called as orthologous gene. There's another gene called as paralogous gene. Those paralogous genes will be arising from a gene duplication event. So speciation means orthologous. S4. You just remember the word S4. Speciation means orthologous and just remember the word DP. What's a DP? D for duplication and P for paralogous. Okay. Next is telomerase is an RNA dependent DNA polymerase. So many are thinking it's an RNA dependent RNA polymerase, but it's an RNA dependent DNA polymerase. Okay. Next, what type of bonds generally stabilize the antigen antibody interaction? So antigen and antibody interaction are stabilized by non-covalent bonds which include van der Waals, hydrophobic, ionic, electrostatic and hydrogen bonds. Okay. Next, Cry-Do-Chat syndrome is a genetic disorder that is caused by the deletion of your short arm of chromosome 5. So you need to remember the word deletion and it is due to a deletion of your short arm, not a long arm of chromosome 5. It is chromosome 5, not chromosome 15. These kind of silly mistakes you will be making. That's why I am insisting you to read each and every line properly. And in my part 1 video, I had made a mistake on question number 27. It is an exogenous antigen. So exogenous antigen are presented by class 2 MHC. Okay. So just do a correction while reading question number 27 in part 1. Next, which intermediate filament proteins is present in nucleus? So it is lamin. So don't confuse lamin with laminin. So this lamin will be coming under type 5 intermediate filament protein. Okay. And it is present in your nucleus. In these kind lamin based questions are asked in CSAR DBT exam. So that's why I have made this particular question as an expected one. Next, cytotoxic T lymphocyte generally recognize antigen that are associated with class 1 MHC. So cytotoxic T cell means CD8 T cell. And your helper T cells are will be recognizing antigen associated with class 2 MHC. So from MHC, you can expect at least 2 questions. Okay. Which enzyme is used to degrade the RNA strand of your RNA DNA hybrid? It is your RNAH. So remember H for hybrid. Okay. Next, precursor for brazinosteroids is campesterol. So the precursor for your jasmonic acid is alpha linolenic acid. Okay, so what is a precursor of your oxide? So you need to tell me in the comment section. Next, selenocysteine is encoded by, sorry for the mistake friends, so it is encoded by UGA codon. And now in the comment section, you need to tell me what is a single letter code for the selenocysteine and pyrrolysine. Next, amino acid with largest number of codons are your arginine, leucine and serine. Each of 3 amino acids that I mentioned over here consists of 6 codon. Okay. Next, codominant marker include your RFLP, SSR, STS. Whereas your AFLP comes under the category of dominant marker. Next, cellulase is an enzyme which is commercially produced by a fungus called Trichoderma viridae. Next, which viral vector is ideal for expressing therapeutic gene in non-dividing cells? So it is your lentiviral vector. Okay, if the cell is not dividing or it is a terminally differentiated cell, means in that case lentiviral vector will be used for gene therapy purpose. Next, Newtonian fluid include glycerol and water. So your ketchup is an example of pseudoplastic fluid. Ketchup, paint are coming under the category of pseudoplastic fluid, which is a non-Newtonian fluid. And this ketchup, if you consider, means why it is called as non-Newtonian means its viscosity depends upon shear weight. So if you upside down, if you done your ketchup means it will not flow. Only if you apply your force, it will be flowing onto your plate. Okay. So that's why it is called as pseudoplastic fluid. In other words, it is called as shear thinning fluid. Next, tDNA of your agrobacterium must be cut out from its circular plasmid for its transfer into plant cell. Which of the following, which Vir gene is responsible for this? It is the role of Vir D1 and D2. Both will function together and they have a nuclease attack. So they will be nick your tDNA and it will be producing a single stranded tDNA from the circular plasmid of your TI plasmid. Okay, the role is done by your Vir D1 and Vir D2. And your Vir C will be playing a role of helicase. Okay, and already we discussed Vir E is a single stranded binding protein. And sometimes they will be asking which Vir gene will be functioning inside the plant cell. That kind of questions will be asked. Agrobacterium is also an important thing. You can't expect simple questions from agrobacterium or even from some other topics. So in these days from GATE exam, they are asking few advanced questions. So you need to update yourself. Don't worry. Almost if you take 65 questions, almost 30 questions you can easily answer in GATE Biotech. So don't worry about engineering mathematics and your bioengineering and other things. Okay, in these days 30 questions are very easy for you. Okay, so don't worry if you didn't prepare problems and sums coming for this engineering, mathematics and bioengineering. Just concentrate on the topic which you had studied at this time point since you have only 9 days more for your exam, so you need to revise a lot. The first alkaloid to be produced or to be isolated in a crude form is your morphine. Okay, and even your vincristine and vinblastine, which are isolated from this Catharines roses, which is employed for anti-cancer, it is also an alkaloid only. Okay. Next, which enzyme used to prevent unwanted self-ligation of your DNA molecule during cloning? It is your alkaline phosphatase. Okay. This enzyme is used to prevent unwanted self-liking of your DNA molecules, the target DNA and vector DNA molecule during the cloning experiment. Next, in Ramachandran plot, the Tarzan angle around the bond between nitrogen of one amino acid and carbon of preceding amino acid is pi angle. What about psi angle? What about omega angle? You need to tell me in the comment section. So next question is lipid wraps are made of. So lipid wraps are made up of cholesterol, glycorapide and sphingomyelin. Okay. Next, a trinuclear repeats in Huntington disease is CAG. So in this disease, this particular trinucleate will be repeated more than 35 times. And this particular CAG will be coding for your glutamine amino acid. Okay. Next, an immunoassay that measures the frequency of cytokine secreting cells at a single cell level is your ELISpot. So ELISpot is the most sensitive immunoassay that are employed for analyzing your frequency of your antigen presented T cell based on measuring the cytokine, number of cytokine producing cells at a single cell level. Okay. So the next, which program is employed for constructing the phylogenetic tree? They are asking which program is employed for constructing phylogenetic tree. There are two programs. One is phylip and another one is mega. Okay, these are the most common bioinformatics program employed for constructing phylogenetic tree. Next, which CA2 calcium sensor regulates synaptic vesicle exocytosis? It is your synaptogamine. Okay, so I had made a mistake while typing. It is synaptogamine. So kindly please do correct it. Next, which motor protein is responsible for retrograde axonal transport that carries vesicle and other cellular products towards the cell bodies of your neuron? It is dynein. So dynein is a minus N directed motor protein and it is involved in the retrograde axonal transport, whereas other protein like your kinesin are involved in your anterograde axonal transport. Okay. Next question is the second type of regulator calcium channel located on membrane of your endoplasmic reticulum or sarcoplasmic reticulum is ryanodine receptor. Okay, so this ryanodine is named because of its sensitivity to a plant alkaloid called ryanodine. So they inhibit the calcium channel. That's why they have named this receptor. The name of the particular alkaloid is given for this receptor. Next, the primary enzyme responsible for the direct reversal of your UV-induced thymidine dimer is your photolyase. So now tell me whether photolyase is present in humans or not. Okay. Next, during pregnancy, which class of antibody can cross placenta and transfer passive immunity from mother to fetus? It is your IgG. So antibody also very important. You can expect match the following type of questions in antibody. Okay. In alternative years, they will be asking questions with the antibody, friends. So kindly please do enough reading about antibodies like immunoglobulins. It's a type of inheritance in which presence of both the homologous chromosome pair in a diploid offspring being derived from a single parent is called as uniparental disomy. There are two diseases. One is Angelman syndrome and another one is Prader-Willi syndrome. So this Angelman syndrome, it is called as paternal uniparental disomy. So person with that syndrome, they have both the homologous chromosome coming from their father. So that's why it's called as paternal uniparental disomy. Whereas if you take a Prader-Willi syndrome, so it is called as maternal uniparental disomy. Okay. So just know more about that particular thing. Next, which RNA is involved in modification and processing of your ribosomal RNA? It is small nucleolar RNA. So don't confuse this SNO RNA with your snRNA. snRNA is involved in the process of splicing. The last question, degree of reduction of glucose. So this is an easy question. Sometimes they will be asking degree of reduction of ethanol or degree of reduction of acetic acid, like that they will be asking. So first you need to know the empirical formula. For glucose it is C6H12O6. Then this is how you need to find out. So valency for carbon is 4. So 6 multiplied by 4 plus valency of hydrogen is 1. So 12 into 1. Valency of oxygen is minus 2. So if you solve the simple problem, you can know the degree of reduction of glucose is 24. Okay, so just know the valency for carbon, hydrogen, nitrogen and oxygen. So you can simply give a correct answer for this question. So that's it. We have completed 100 questions. So if you still want any help for GATE Biotech, please do tell me. So thank you friends, thanks for watching this video.